Okay, so for this audio lecture, we'll be looking at the development of T cells, T lymphocytes, and this is covered in Chapter 7 of your textbook. Just be aware there's going to be a couple points where I disagree with the textbook or how it's presented in the textbook. And so remember, I write the exam, I grade the exam. And T cells are my thing. All right, so when we were looking at the precursors for T cells, they're going to first begin development in the bone marrow, and then they're going to leave the bone marrow and go to the thymus. And so when they enter the thymus, they're not T cells yet. They need to go through stages of development. And they actually, when they enter into the thymus, they actually have the ability to become other cells, like DCs, macrophages, natural killer cells, and such. So just keep in mind that the cell that enters into the thymus is not destined yet to become a T cell. It's the process it undergoes in the thymus that allows it to then be destined to the lineage of T cells, and whether that's a CD4 T cell or a CD8 T cell. After the T cell matures, it's going to leave the thymus and then it goes to secondary lymphatic tissues where it can become activated if it's presented its antigen. Just some basic, what does the thymus look like? So here you can see some of the structures of the thymus. You can notice in the blue area on the right-hand figure, that is where your cortical epithelial cells are going to be. So that becomes important. Where the red cells are towards the bottom, that's your medullary epithelial cells. And again, that becomes important because those epithelial cells play an important role in presenting self-antigen and MHCs to the thymocytes during their development. So when we're looking at production of T cells, in your beginning years of life, that is when your T cell development is most active. And so that's actually when your thymus is the biggest. And so as you age, your ability to produce T cells decreases with age. Okay, so keep in mind that this development stage is most active in young individuals. As you age, the thymus gradually is replaced by fatty tissue, but it does shrink to some degree. When we look at the commitment to the T cell lineage, there's a number of proteins that change. So just like B cells, T cells are going to change protein expression during their development. So this is just looking at some of the different adhesion molecules, co-receptors, and their expression level as the T cell is going through early development. So this is going from an uncommitted progenitor cell, so like a lymphoid progenitor cell. And you can see how the stem cell surface marker is expressed. We have one adhesion molecule being expressed, but when the cell becomes a thymocyte and it's double negative, and we'll talk what that means, then it changes the expression. It loses that stem cell receptor, and then it starts to express other adhesion molecules, cytokine receptors, and also RAG, right? So it needs to be able to undergo recombination events to create that T cell receptor to make it a T cell. So during the process, there's a number of different markers that we could look at. Some of the common ones are c-Kit, so similar to B cells. T cells, early thymocytes, are going to also express c-Kit, so it can bind to stem cell growth factor. It's also going to express CD44 as an adhesion molecule, and then it increases expression of CD25, which is the IL-2 receptor. So IL-2 is very important in the development of the T cell, but also later on in the activation of the T cell. And then obviously the T cell receptor also is going to undergo rearrangement during the development. One of the critical markers that are looked at is Notch1. So Notch1 is known as the commitment marker. So when a common lymphoid progenitor is expressing Notch, that indicates it's going to go to the T cell lineage. So if you remove the ability to produce Notch1, it will not be able to commit to becoming a T cell. It will not become a T cell. So with expression of Notch1, we also get GAT3 transcription factor being activated, and that causes a whole bunch of signal, pathways being activated and transcription of different genes that allow for, expression of those proteins that are critical for the T cell rearrangement events. So keep in mind that T cells can develop two different receptors, so they can have the alpha beta receptor or gamma delta receptor. So here we have our uncommitted progenitor, so our common lymphoid progenitor. It is going to change expression of surface markers. That starts to signal for expression in rearrangement events to the T cell, so that it's going to express either the beta chain or gamma and delta. If it expresses the beta chain, then alpha is going to get expressed too, so that T cell will then have an alpha beta receptor, as you can see here. And so once that receptor is made, it's committed to that receptor type. And one of the terms you'll see here is double negative. So sometimes that's abbreviated DN. So if you see DN, think double negative. And what they're referring to is it's negative for CD4 and CD8. So there's going to be times where the T cell during its development is referred to as double negative, double positive, and single negative. So just, again, we'll talk about that in a little bit later. But it's during that double negative stage that the recombination events for the T cell receptor occur. And so, as I mentioned, there's two options for the receptors, if you'll remember from last unit. So there's either alpha beta or gamma delta. And so generally beta first gets expressed. So the majority of your T cells have alpha beta, but they can have gamma delta. So again, it's during that double negative stage, which we have these rearrangement events happening. So we're either going to get expression of beta or gamma delta. And so if we have beta, we have a pre-T cell receptor that's formed, and then we're going to get expression of the alpha chain to match with it. All right, and so just like the B cell can try to rescue a non-productive arrangement, a T cell can also do that. This is where I have a disagreement on how this is presented. So kind of ignore the figure you're looking at and think about gene segment recombination for the T cell. And so again, just like the B cell, where we can select variable regions that are still available to they're still available on the gene after our initial resegment combination event that leaded a non-productive arrangement, we can use the segments that are left to try to create a productive beta chain, okay, that would lead to a productive functional T cell receptor. So again, they can undergo two attempts to fix and correct it. Again, I just don't agree with how this is presented as far as the segments that are left. At the same time, if the alpha chain is non-functional, is non-productive, they can go through additional attempts in order to try to rescue that by, again, picking out different gene segments for expression. This figure I agree with more so in how it's depicted. Okay, so just keep that in mind. And again, the whole idea is that you're going to be taking RAG1 and RAG2, holding those, the DNA in position so that cuts can be made, so additional segments can be removed. And again, in hopes to yield a productive T cell receptor. Just like B cells, not all T cells are successful at doing this. So if they fail to rescue their T cell receptor through these additional rearrangement events, then that would mean that the T cell would go into apoptosis. So just like with B cells, they're going to change expression of proteins to reflect the processes that they are undergoing during the different stages. So you can see here in this figure, we have our double negative T cells that are developing their pre-T cell receptor. And so in order for that to happen, we have to have expression of RAG1 and RAG2, right? So recombination events can occur. We can also see exchanges of expression of additional molecules. We can see Notch1 has come up and continues to be expressed, showing that T cell commitment. And we can see a number of signal transduction pathways and transcription factors, because these cells are very active in taking that DNA, splicing the DNA, expression of the T cell receptor genes. Okay. Just to point out, this is another area where I kind of disagree with how the textbook's presenting it. In order to enter into double positive stage, we've already had the rearrangement events of our accompanying T cell receptor gene. So if we're talking about beta, then alpha has already, in this textbook, it's showing that during early double positive events, you're having expression of that. And again, I disagree with that. So just be aware of that. And that comes up in the next figure. All right, and so as the cells are migrating into the thymus, you can see in the figure in the bottom, we have these progenitor cells migrating in. They are going to get support from stromal cells, these epithelial cells that are going to produce chemicals, cytokines, chemokines that will help, and factors that will help in their proliferation and also maturing through these processes and these stages. Initially, these T cells, one of the first events is committing to the T cell lineage. And so you can see as they're entering into the cortex, in this double negative stage, that's where they're going to commit to T cell lineage, and that means that Notch1 is being expressed. They're going to undergo rearrangement events with the beta gene so that they are expressing the pre-T cell receptor, and this becomes a checkpoint. The cells are still at a double negative stage, and this is where I disagree with what's presented in this textbook. Rearrangement events of the alpha chain are going to occur, and then they become double negative immature T cells at that stage. There's additional checks on the T cell receptor to make sure it's functional, and also that it undergoes what's known as positive selection and negative selection to make sure that, again, that T cell has a good receptor. It's going to undergo positive selection while it's in a double positive state. It undergoes negative selection when it's single positive. So when we're talking about positive selection is when the T cell is checked to make sure its receptor can interact with an MHC. Okay. And again, this is going to occur when the cell is double positive. So when we're talking about double positive, it is going to be positive for CD4 and CD8. Okay, so they're referred to as DP thymocytes. And this is actually what makes up the majority of the thymocytes that are in the thymus, are in this double positive state. Most cells actually fail positive selection. So the T cell receptor that they have went and produced through all this process actually can't interact with an MHC, or it interacts too strongly. So what you want is a T cell receptor that interacts with the MHC just right, Goldilocks, right? And the reality is that about 95% of them fail. And when they fail to undergo positive selection, they fail to receive survival signals, so they go into apoptosis. So you can see here at the bottom, if the binding is weak or there's no binding, the cell's going to die. Now, if this is again where I disagree with the textbook, they have it that if there's moderate to strong binding, they both survive, and that's not correct. So I disagree with that. So if there's strong binding to to the point, where MHC and TCR is almost locked together, that actually is not going to allow for it to survive. So it's actually going to go into apoptosis. Okay. It's the cortical epithelial cells that are responsible for this process. So again, keep in mind they're towards the outside of the thymus, and they're going to express MHC class 1 and class 2. And so they present tissue-restricted antigens, or TRAs. And so these are antigens, these are proteins that are only going to be expressed in the thymus for this purpose, if you will. And so it's not self-antigens in the sense of antigens that would be produced outside of the thymus. And this is a very unique function, right? So when these MHCs have these peptides loaded on MHC class 1 and class 2, it really, what we're checking for is, does the T cell receptor bind to, is it able to interact with the MHC? Because again, keep in mind, T cells have to be presented to. So if these T cells can't interact with the MHC, we don't want them to survive because they're not going to be of any purpose out. We don't want to expend any more energy on them. So again, T cell receptors that can't bind are not going to get survival signals, so they actually die by what's known as neglect. T cells that bind too strongly are going to end up undergoing apoptosis. This is not, so the term gets used, negative selection. It's not negative selection as a process. It's you are not positively selected. So it's the lack of positive selection. And this textbook and other textbooks will call it negative selection, which just leads to confusion. We'll talk about what negative selection is in terms of autoimmunity in just a moment. And really, again, looking back at positive selection, what we're looking for is the just right? The Goldilocks. The T cell receptor and the MHC interact with the perfect affinity that's going to allow them to interact and share information, and for the T cell to become activated, not in the thymus, but has the potential to become activated out in secondary lymphatic tissue. That's the one you want to go on. So that's the one that's positively selected and is allowed to survive. So in this textbook, they present one theory on how T cells then move to a single positive state. There are other theories that I will talk about in the next slide that is from a different textbook. So I just want to caution that the textbook presents this as like, this is fact, and it's one theory. So one theory on how the T cell knows which single positive to become, because when they're single positive, they're going to express either CD4 or CD8. One of the theories is that depending upon what the receptor can interact with, that will determine what type it becomes. So what's shown in this figure is, here we got these double positive thymocytes. So you can see our T cell receptor, and we can see CD8s, but if you also look, there's also CD4 being expressed on this upper left panel. It's going to interact with the thymic epithelial cell, and because that T cell receptor is interacting with MHC class 1, which is used by CD8 T cells, it's going to become a CD8 T cell. On the other side, we have our double positive thymocyte, and it's interacting with its T cell receptor with an MHC class 2. So the co-receptor for that is CD4. So CD4 T cells interact with MHC class 2. So this is going to become a CD4 T cell. Okay. So this theory, this model states that during the positive selection state, when it's double positive, which MHC it interacts with commits it to either a CD4 or a CD8 lineage. And I'm not saying that this is wrong, I'm just saying this is one of the proposed theories, and the way it's presented in this textbook makes it seem like this is the theory. So the other theories are that there are cues and signals, or is known as an instructional model, is that there's cues and signals that are generated that instructs the T cell to which lineage. So this is most similar to what we just saw, right? So the cues would be because the receptor is interacting. We also have the systolic model. So this is where what happens is through positive selection, it's just randomly one will get downregulated. So it's not because of interaction with an MHC versus 1 versus 2. It's just a random, like, okay, 50% of you are going to downregulate CD4 and 50% of you are going to downregulate CD8. So you're becoming the other one, right? And then those cells then give their function, right? So again, it's this randomness. The other one is kinetic signaling molecules. So this is where it really depends upon the cytokines that are present. And so with this theory, with this model, T cells are going to commit to the CD4 lineage if they receive a continuous signal in their commitment during positive selection. Where CD8 cells will commit to that lineage if the signal becomes interrupted. And this is where we know that CD7 promotes CD8's differentiating. And so the work that's been done on the kinetic signaling molecule really kind of contradicts what's being presented again in the last figure as like the truth. Okay, so again, just remember that further investigation needs to be done to understand why T cells, when they become single positive, will commit to one lineage over another. How do they know to become CD4s and CD8s? And maybe the data will hold out. Maybe, previous work that's been done, by furthering that information, we'll find that one of these models is the one. The reality is probably a mixture of all of these, right? So there's probably some kind of like override. So maybe the MHC that it interacts with really dictates it. But if in the environment we need to have more CD8s and we have IL-7 present, then that's going to push to CD8s, right? So it's not a one-size-fits-all model kind of thing. All right, here's my big issue. So this use of negative selection as a term, like this figure is not talking about negative selection as the term negative selection that we're going to be talking about in a moment. This is talking about the lack of positive selection for these cells. Okay, so my other issue with this is the same figure as 716, except it's a different color, and instead of the epithelial cells, it's showing dendritic cells presenting. During positive selection, you can have other cells presenting MHC class 1 and class 2 to these thymocytes. So again, do not confuse what is labeled in this figure as negative selection, the process that leads to tolerance. Okay, so for my purposes, if I was writing this textbook, I would have omitted this figure because, again, you're talking about positive selection in figure 716. Again, not completely presented to where I would agree with how it's presented, but that was a whole lot better than adding this additional figure to like just further confirm and confuse students. So keep in mind, if I'm asking about negative selection, I'm asking about negative selection related to central tolerance, okay? So negative selection is part of central tolerance. So central tolerance is what's happening in your primary lymphoid tissues. There's also peripheral tolerance, so tolerance that's going to occur once your thymocyte has left primary and gone to secondary. But central tolerance is critical to self-tolerance. Self-tolerance is where you do not respond to self, okay? So your T cells and B cells undergo negative selection. Negative selection, a process to make sure that they don't recognize self. Again, that's very different than that word combination of words they're using in that previous figure, that previous slide. Okay. So during negative selection, for T cells, we've already undergone positive selection. So now we're going to actually present self antigens to the T cell. And again, we talked about this process with B cells. So that we're going to kind of try to trick the T cell, if you will, into responding. And if it does respond, it's going to get deleted. Okay. So clonal deletion will be induced, which is just fancy way to say the cells are going to go into apoptosis. Okay. So if the T cell sees self-antigen, if its receptor is able to bind self-antigen that's presented on MHCs, it's going to be told to to undergo apoptosis. Now, how are we having expression of and processing of self proteins? It is through the AIR protein. So autoimmune regulator or AIR protein actually induces the expression of proteins by the medullary thymic epithelial cells. So as that T cell goes back into the medullary tissue, so leaves the cortex, it's now single positive, it's going to interact with the epithelial cells there, and the epithelial cells are going to be producing proteins. So it turns on, remember that these cells have the whole genome, and so it turns on expression of proteins and then uses the proteasome in order to degrade them and then present them. So this allows for the presentation of all these fragments that the T cell could potentially see out in circulation, and in a cell that does recognize that is going to be told to undergo apoptosis. A cell that does not recognize self-antigens, so it has a negative response, no response, it is allowed to survive, and it gets cues to then exit. So one of the, when we're trying to tell the story and make it so the stages are kind of linear and like, okay, cell goes here, then goes there, then goes there, then goes there, it's easier to memorize that, right? And so keep in mind that it's likely that in some cases negative selection can occur at various points of development. So while positive selection is occurring, where we're checking for the MHC and T cell receptor interaction, then, it's possible negative selection is also occurring too. We do know that positively selected cells end up expressing CCR7, which is a chemokine receptor, and that tells them to move into the medullary, and that's where negative selection is going to occur because it's in the medullary where you have those thymic epithelial cells that express AIR that will then express self-proteins. Okay, so remember in the cortex, it's a limited protein expression that are tissue specific that get loaded onto the MHC to check for positive selection. Okay, so again, it's always more complicated than what we make it out to be. So keep in mind that thymocytes do have the ability to become other types of lymphocytes. So even the double positive, so the common lymphoid progenitor, when it's entering in to the thymus, it has the ability to become all these different other cells. But even at the double positive stage, they can become T cell-like cells, like natural killer cells. Also intraepithelial lymphocytes, so these are usually CD8 cells, but they act more innate-like than what we see with our traditional CD8 cells. And then we can also get regulatory T cells. These are CD4 subsets. So if we kind of think about it, CD8 and CD4 each have these unique different functions that they could have if they don't become your traditional CD8 T cell or CD4 T cell. We'll definitely end up talking about T regulatory cells later on in the semester when we're talking about activation, because they play a critical role in helping with tolerance in the periphery. They are kind of like the nannies. So just a couple summary slides. So I put a big X here because I disagree with what they're calling negative selection, because really what they're outlining here is the negative selection of positive selection. It's not really talking about true negative selection in the sense of tolerance. So do keep in mind that really that third row really needs to be revamped if they want to call it negative selection. So negative selection, so if I was to revamp this so this was negative selection, the thymocyte location characteristics would be single positive in the medullary, interacting with medullary thymic epithelial cells that are presenting self-peptides, fragments of self-proteins. I kind of like this image a little better. So this is from a different textbook. So lots of great information on here. maybe you'll like this one better than some of the images in your textbook. So again, keep in mind that we have these precursor cells that are coming in. They're going to be double negative to begin with. And as they commit to the T cell lineage, they're moving out to the cortex. So first event that's going to happen is that they're going to commit to the T cell lineage. This is usually referred to as the DN2 stage. This information kind of is lacking from your textbook. Keep in mind that this early double negative stage, they have the potential to become other cells. So they can even become myeloid cells or natural killer cells. That's really important because we need presenters in the thymus to undergo negative selection, right? So and to also potentially do some positive selection where they are interacting with potential presenters and MHCs later on. During what's known as the double negative 3 stage, this is where we have the T cell gene rearrangement. So first expression of beta and then alpha. Again, I'm in agreement with this textbook more than our textbook for this chapter. And then it's going to go into a DN4 stage, and that's just really a pre-double positive stage. Then the T cell is going to be double positive, so it's going to express both CD4 and CD8. During this time, this is where it undergoes positive selection. So positive selection is going to determine whether it can interact with MHCs. So if the T cell receptor is not good, if it lacks positive selection, then it will go into cell death. If it passes that positive selection, it's going to become single positive. Again, different theories on how that comes out, how that T cell knows to either become CD4 or CD8. Then it's going to undergo negative selection. So during negative selection, it's going to interact with self-peptides because of the expression of air. Air allows for the genome to be turned on, proteins to be expressed, and then processed through the proteasome, and then presented to these immune cells, to these T cells, so that the T cells that would recognize self are going to undergo apoptosis and be deleted. All of that is also outlined here. This is also from a different textbook. Keep in mind, again, I don't know why they use this term negative selection when we're talking about positive selection. Call it the lack of positive selection. I'm going to write that textbook now. So in the cortex, what you're seeing labeled as negative selection is not negative selection, is the lack of positive selection. So your options in the cortex while the cell is undergoing positive selection is that you have positive selection, you have death by neglect, or you have the lack of positive selection. Okay. And then again, this figure nicely shows how we're going to move to the medullary tissue. We're going to have air expression. That's going to allow for the thymic epithelial cells to express fragments of self-proteins on MHCs and present to CD4 positive T cells and CD8 positive T cells. And if they interact, again, it's kind of a nasty trick. If they do interact, they're going to go into apoptosis. If they don't interact, they're allowed to survive, and then they can migrate to the periphery. They can go to secondary lymphatic tissues. I should have mentioned the medullary thymic epithelial cells. They express both MHC class 1 and class 2, so they're kind of unique. They're given that privilege because of that AIR expression. At the same time, you can have other immune cells like dendritic cells also presenting, as you can see here in this image. All right, if there are any questions, please feel free to contact me so that we can go over those during class time. Again, this is one of the chapters that I have some disagreements on how it's presented in this textbook. And so, again, I'll hopefully in this audio lecture, I stressed those differences. I will make sure in class time I also stress those differences so that, and I also go through inquisitive questions and look to see if there's any that I disagree with so that you're aware of those. And that, again, you have a fair understanding of what my expectations are when it comes to assessment pieces. All right, so have a good day. Thank you.